Welcome to Halteres Presents, a brand-agnostic interview podcast that seeks to objectively highlight the happenings within the world of diagnostics. And now, your hosts, Rich Thayer and Mickey Urdea.
Welcome to Halteres Presents. This is Rich Thayer, Managing Partner at Halteres.
And this is Mickey Urdea, the founding partner.
We're very happy to welcome back Dave Engelthaler to this podcast.
We had a great conversation with Dave, as usual. He's extraordinarily knowledgeable about avian flu, the origins of it, the implications, surveillance, treatment. It was a fascinating conversation. I think you're going to enjoy listening to it.
And now, our interview with Dave Engelthaler.
Dave, thanks so much for joining our podcast. We're really pleased to have you back as our guest. And please tell our listeners a bit about yourself and the work that you've done at TGen and at ASU now.
Yeah, thanks, Mickey. It's great to be here and an honor to be back. So Dave Engelthaler, I'm the executive director of TGen North, which is the pathogen and microbiome division of TGen, or the Translational Genomics Research Institute. It's a nonprofit research institute that really spun out of the Human Genome Project back in the late 90s, early 2000s, to really, how do we translate all of this great new information that we're getting out of the genome into knowledge and into action around chronic health conditions, infectious disease conditions, neuro disease conditions as well. So better diagnostics, new novel drugs, vaccines for a whole host of issues based off of genomics. This was in the early days of genomics that we were founded, so a little over 20 years ago, and now is really now become the mainstay of all of biosciences in using genomics. So that's been a lot of fun. My group has been mostly focused on the infectious disease side and the public health component. And that's primarily because my background has been in public health and epidemiology for the past 30 plus years. I was the state epi at Arizona. I worked at CDC and did a lot of on the ground public health practice and a lot of now public health research. And then most recently, a little over a year ago, I've also taken on the hat of being the executive director of something called the Health Observatory at ASU, or Arizona State University. And this is, again, the whole concept of translating health data into health knowledge, but across the whole human healthscape. We're really looking at all the different sources of data around health. So you can think about clinical health data, you can think about the population-based health data, thinking about vital records, social determinants of health, environmental health, bringing all this together with advanced analytics, AI tools, advanced modeling to generate new knowledge and then be able to get that new knowledge out to the people that need it, whether that's healthcare officials, elected officials, the general public. We all need better health knowledge that's more precise to the conditions that we're having to deal with. And that's a big part of our goals.
So Dave, avian influenza has been in and out of the news for the last few years, but of course it's been in and out of the news for about 100 years now. Can you please tell our listeners a bit about the origins of avian flu?
Yeah, we continually are concerned about influenza because we've known that it's caused these massive pandemics in the past, that we still see flu every year among humans to a degree where we have tens of thousands of deaths, even just here in the US. So flu is a really important infectious disease that we really have to deal with on an annual basis. Avian flu is essentially a series of strains of influenza that dominantly infect birds, and birds can move the virus around. And sometimes we have something called highly pathogenic avian flu, which will just wipe out entire flocks of birds, whether it's wild birds or poultry, commercial agriculture birds. And we've been seeing that really up closely for the last, let's say, 30 years happening. Occasionally, you get this spillover into humans. And what's really been concerning is the strains that we were seeing in Asia, as well as in the Middle East, going back 20, 25 years, have, when they get into humans, we don't ever see human-to-human spread, but we see incredibly pathogenic and virulent flu in humans, so much so it's about a 50% fatality rate. So if we think about a flu with a 50% fatality rate, that's the stuff of Hollywood and nightmares. So we are very concerned about it, but thankfully, avian flu has not become a human-to-human spread virus in all its different forms, including the most recent forms that we've been seeing here in North America.
I mean, you dropped a bombshell there. I'm not quite sure how to phrase it, but 50% mortality rate. This is in the spillover cases, I guess you're referring to here.
Yeah, and I want to emphasize too that the strains that we have been seeing in North America over the last couple of years don't have that high fatality rate. It's actually been pretty mild most of the time in humans, which is also good news. But we're always worried because flu does keep changing in a number of different ways.
Yeah. And it's not going to the receptors we most worry about it going to, right? I mean, that's really the issue, isn't it?
Yeah, so with avian flu, it's still an influenza virus, but it does have some different components that allow it to more readily infect birds than humans or other mammals. And one of those is that it will attach to different cell receptors. So we think about the sialic cell receptor. There's an alpha 2/3 receptor that birds commonly have, and avian flu viruses really are able to bind to those and then infect cells really easily in birds. Humans and mammals dominantly have something called the alpha 2/6 receptor, and the avian flu virus does not infect those very well. It's not like the right key fitting into the lock. But we actually, humans and mammals, do have that bird receptor, that 2/3 receptor in different places. And one of those places that it occurs is in the eyes. We actually have those 2/3 receptors in the eyes, which is why most of the flu cases, avian flu cases we've seen in humans over the last couple of years have really had these blood red shot eyes, this conjunctivitis with really bright red eyes. As well as in our gut, we have those same receptors in our gut. So we can have these gastrointestinal infections as well, but really not so much in our lungs, especially the upper respiratory component of our lungs. So we don't typically see the virus infecting us like other flu viruses do. There's one caveat, which is down deep in our lungs, we do have those 2/3 receptors, which if the virus were to get down that far, occasionally we have severe cases. It was more common in the older Asian lineages that we were seeing, going back a decade or so, that we would get the lower respiratory. But with this version we're seeing in North America, it's pretty rare to actually have those kind of infections.
We've heard, Dave, that there's some concern about mutations which could occur and would make it go from the birds to the humans to human transmission. Is that something that people are keeping a close eye on?
Yeah, one of the things that we've always been concerned about with influenza viruses is that it evolves in a couple of different ways. Uh, in some in very kind of subtle ways, what we call antigenetic drift, where you just picks up mutations here and there, like all viruses do, and sometimes those mutations can really make a difference. Like it allows the virus to infect a cell quicker, or it can maybe have a different pathology effect once it's infecting a cell. But the other way that it evolves is that the virus has what we call antigenetic shift, where you actually have these major different genetic components of the virus can actually interchange with other viruses. So if you get an individual, an animal infected with two different influenza viruses, those viruses can recombine essentially. You get like a recombinant virus, almost a Frankenstein virus, and then you get something new. Most of the time, that's going to be a dead end, not really good at getting out of the host or causing new infections. But every once in a while, you get a version that actually does spread easier or becomes, more virulent, or in the worst case, is that it becomes both.
Sure.
Thank you, Dave. That's certainly good food for thought here. The most recent scare with avian flu has involved the infection or spillover, as you say, to dairy cows. But interestingly, no infections appear to have been reported in beef cows. A few people have contracted avian flu by working in the milk industry, as we've just discussed, mostly as eye infections, but we've not heard stories of people outside the industry becoming infected too. What's going on here? And has the present danger from infected milk cows subsided?
Yeah, there's a lot of great questions in there, Rich. So let's maybe start back a little bit. When the strain that we started really seeing in North America in 2022, late 2022, when that strain essentially had moved around the world and became a pandemic strain in birds, is because it picked up a few mutations that allowed it to really transmit easily among birds, more easily than it had been. So then migratory birds took it around the world and it showed up in the US. That version didn't seem to readily infect other mammals, especially what we were seeing in cattle, except when you may have a predator mammal that eats an infected bird, then they can get infected. But then there's not a lot of transmission among those mammals. Then we started to see some of this, what I was calling antigenetic drift. Some new mutations occurred, and then we get this new, what we're calling a subclade D.1.1. Now, there's a lot of terrible names for these viruses, so I'm going to try not to use too many of them. But this new subclade seemed to actually start to infect cattle more easily, and it spread even more easy in birds and has now become the dominant version that we see. Interesting, though, was that cattle were getting infected. They could transmit cattle to cattle more easily than we were seeing this virus being transmitted in other mammals. One of the things is that cattle have a lot of these alpha 2-3 receptors. So like the avian flu receptor for birds, they have in the mammary glands, so that when the mammary glands get infected, the milk also becomes infected, and they essentially excrete a lot of the virus out through the milk. So then you get a lot of this virus out in the milk, and then milk handlers in cattle industries, we're getting exposed to a lot of the virus, and it's not the cleanest operation. You get it in your eyes or other ways that you could get infected. So we're seeing human cases that way. That's why we're getting a lot associated with dairy cattle, say, versus beef cattle, which they would probably, if they get infected, they would probably excrete it in their milk as well too. But that is not the main point, and all the exposure is not happening around the mammary glands of beef cattle. What's also been very interesting is that's how we've seen this show up in other animals, is from dairy milk. We have milk that is essentially in farmhouses or at dairy operations that is just given out to the local cats. A lot of these places have cats around to keep the mouse populations down, but the cats were getting infected and then dying of avian flu because cats were just taking in so much virus from this infected milk. Then we were starting to see cat to local animal population transmissions as well. So this whole getting infected, this new kind of version that became adapted to cattle, allowed it now a new way to get into different mammals. And then, of course, then the risk is, can we get it from milk as well? Right? If cats can get it from milk and cattle can share it back and forth, can we get it from cattle milk? And the answer is most likely. We actually have not seen very many human cases. Actually, only one suspect case from actually drinking infected milk that wasn't pasteurized. But theoretically, it's there, and there's a lot of virus there. This is why we just reiterate the public health recommendations of only drinking pasteurized milk. One of the other interesting things where we've been seeing a lot of cases in cats is not just from infected milk, but from this raw cat food. A lot of different producers make raw cat food and raw dog food, and they've been using and unfortunately have included infected birds in the creation of that. So then you start to have multiple cats dying from this brand or that brand of raw cat food. So we do have to be on the lookout for that and new ways that we end up getting exposed that we weren't actually ready for before. So we just have to keep adjusting our priors and coming up with increasing our our not just our innovation, but our imagination as to where and how this virus and future viruses may affect us.
Wow. Is that well known to vets? I mean, are they warning their clientele about that?
I see a lot of it in the literature, but I couldn't tell you as far as a knowledge survey on vets.
Mm-hmm.
So just thinking about then, is there ongoing risk? We know about this. There's a lot of mitigation efforts to try to really screen for this in milk and then determine which dairies may have this virus. And then you essentially end up just dumping all the milk that's infected, even though you can pasteurize it and treat it just fine. But the whole point is, let's try to have public health mitigation efforts every time we do detect it. And by doing more surveillance, we've limited the amount that we've been seeing in dairy cattle. which is good. But we've also seen a kind of a downswing in total virus being transmitted around the country, even in wild birds. That may be on the increase again. We saw it increase last fall. Probably has to do with birds moving around, the migratory patterns of international migratory patterns of birds. We are likely to see more and more coming into the fall. That means probably more exposures of birds to cattle operations, which might mean we'll see more of these types of cases occurring in dairy cattle. But at least we're operationally much more aware now. We are gaining more intel by doing more surveillance, but we don't have a system to completely prevent it.
Yes, I've noticed this fall a total change in the raptor migration here. I live in Idaho and never seen them, hundreds of them at a time coming through. And this is where a huge amount of dairy operations are. So it just makes me wonder, what has changed? I mean, last night we had about 150 turkey vultures circling over the property here and a huge kettle of them. Never seen that before. Saw the same group 2 days ago, and all the dairy cows, right? They die. These cows die from flu. They're out laying in the field till they get picked up. So one would envision the transmission that you're talking about.
Yeah, it's certainly possible. I haven't heard any reports on changes in the migratory patterns this year, but I'm also not following it very closely. So
Yeah.
I don't know about the migratory patterns, but we do know that the virus has been affecting bird populations. Thinking, Matt, even let's just switch to another virus real quickly. When West Nile virus came through in North America back in 2001, as it's going across North America, in some cases, it was wiping out entire bird populations. The corvid population, which is the ravens and crows in Illinois, almost got completely wiped out, except for relatively few that became a bottleneck population that may actually become immune to that virus. But it really substantially affected the overall bird populations. We're seeing that with avian flu too, where we can have decimations of maybe relatively small specific, say, raptor populations or other populations, that it does change the dynamic of bird ecology. One quick example I got to point out is one of the first ways that we saw it showing up in Arizona was that it was killing the California condors. This is a species that was essentially almost extinct, brought back this tremendous heroic efforts to bring this great bird back around the Grand Canyon and Vermilion Cliffs in northern Arizona. And then a couple were found dead. Because they track them all really closely, and it turned out it was from avian flu. It's because these birds, like the turkey vultures you were talking about, they eat dead and
Yeah.
downed birds and other animals, so they get the virus really pretty quickly. What was also heroic was that groups went out, were able to retrap all of the condors and vaccinate them with essentially experimental vaccine, which seems to have held, and they haven't been documented any other deaths from avian flu in the condors. So that's great, but it easily could have wiped out a pretty small population had that not occurred.
Yeah, yeah. I coincidentally am aware that my wife and I fund part of that effort because most of those condors were raised and released from Boise here, from the Birds of Prey Center. So let me ask about the apparent recurring or cyclical appearance of avian flu in the poultry flocks around the country. And might we expect the same thing sort of to happen in the dairy herds? Would it correlate together, or there may be other factors that could create that recurrence in the dairy cows?
Yeah, I think when we see it showing up both in poultry operations and birds as well as in dairy cattle, it's not because the virus has just been there lurking and then it reoccurs. It reoccurs because new infected birds come in to those operations. And if we do see, a continual transmission among wild birds, and then they show up in these different operations, it's quite likely that we're going to continue to see these recurrences, even in places that recently culled all their animals, which they try not to do that with the dairy cattle. They do that with bird operations, and we've seen that in egg layers and in broiler operations, where then they have to cull the whole flock millions of birds and tens of millions of dollars, and then they repopulate, and then it comes back again. And that's just because we don't have big giant nets over these operations that would keep out all wild birds as they fly through. So it's really hard. Biosecurity is really hard to do in keeping infected animals out. So I got to point out, there's one thing that really we saw, like with the condors, that worked really well: vaccination. And in Europe and other places, they do vaccinate their poultry to prevent them from getting sick. We don't do that here, which is because of economic agreements around us not vaccinating our chickens, whether and typically the broilers, because then if they're vaccinated, they can't be sold overseas. And that's prevented us from vaccinating even our egg layers, which would never be sold overseas. But there are some policy problems that is preventing us from actually really getting the vaccines out to preventing this from happening in poultry operations. On the cattle side, we're still working on having good vaccines there, and then also on the human side too. We don't have any really good vaccines that are available for humans yet.
Very interesting. Thank you.
So we've talked about poultry and we've talked about cattle. We know that it gets into a lot of other animal populations. Is there anything being done to monitor other populations concerning spillover events that could occur?
Yeah, there are in a couple of different ways, and some that actually my teams are involved with too. But let's start on one side. Another agriculture animal, which is really important, is the swine population. So pigs, obviously pork, are really important when we're talking about flu and the potential for new variants to occur. And that's because pigs are really the one animal that can become equally infected with both bird flu strains and mammal flu strains, and because of the receptors that they have. And if you can become infected with both, it is quite possible that you have a recombination event occurring in an animal, in a single pig, that gives us a new strain that actually takes on the worst of both: easily transmissible but infectious to humans and pathogenic to humans. We see this as the potential for a start of a new pandemic. When we had the pandemic, the 2009 H5N1 pandemic, everybody remembers that. That was the first pandemic of the millennium. That was coming out of primarily agricultural operations in Mexico, which likely included swine and recombination of multiple flu viruses within swine. So we do have to really look at that, although it doesn't seem to be a big player right now in the current H5N1 situation in North America. That is something that's being watched closely, very closely by USDA, and then others on a local level. Other animals that we look at, there's different surveillance capabilities out there, and so we work here in Arizona. We work with, say, our game and fish department when they find suspicious dead and down animals or birds. The collective research community supports that and does testing to look to see if we find H5N1. We have found some in samples that were submitted, or samples actually that we've collected as well, of wild birds found, say, like in a local park pond, that were dead. So we've been able to find it that way. And by doing that, we're just getting a little bit better eyes on where the virus is. We're also working in a place that's been really highly impacted, which is zoos. Zoos have to continually to do monitoring and surveillance for any sick animals, because avian flu has had a tremendous impact on not only the wild bird species that they have in the zoos, but also some of the wild animals, especially the big cats. It has killed off a number of the big cats, the tigers and the lions and the mountain lions and others, which are really susceptible. They eat a whole infected bird that maybe fell into their pen, get a really bad infection, and then become susceptible and die. And that's, we're talking tens of thousands or hundreds of thousands of dollars per animal. So real big economic impact too to zoos. So here in Arizona, but elsewhere, zoos are really watching this fairly closely and have different types of surveillance in place.
Do people attempt to keep pigs and cows away from one another because of the concern?
It's less so about pigs and cows and more about pigs and birds. But oftentimes we see that, yeah, they're in close proximity. People have a backyard farm. And in one case in the Pacific Northwest, I believe a year ago, that we did found a pig infected with H5N1, and there were also poultry birds on the premises. It was like a backyard type of an operation. So that does happen, and that is a concern. But it's also well known that you have to keep that in the large agricultural settings, and they're typically not raised together because of the easy transmission of virus between them.
Are there other types of testing or surveillance that we can be doing that's not being done now that we would suggest?
Well, what's not being done now is more testing. Like we're really only have, I'd say, a minimal view on this virus and in the different ways to look at it, whether it's in agriculture or we're looking at wild animals and birds, zoos, other places. I think we do need a concerted effort to really increase the overall surveillance. We have a lot of capability in this country, which we saw actually during the COVID pandemic, that can be brought to bear. Not that we have to go into emergency response mode right now, but let us set up systems where we are really improving our overall intelligence gathering on this virus. Because once we start to not see human cases or there hasn't been dairy outbreaks, we start to forget about it, and then all of a sudden we start to see a lot more than we expected. This happens with all sorts of infectious diseases. So I am fearful we don't have enough of that type of surveillance happening across the country in an organized manner. This is part of the problem. We don't have great organized nationwide surveillance. And so we have pockets of great surveillance happening and then lots of gaps throughout the system, which means that we really don't have good intel overall. So I think we do need more testing, as well as continual genomic analysis. We really did this again during the pandemic, started watching like all the variants that were happening, and in some cases really important clinically, because you couldn't use this particular monoclonal antibody anymore because of this variant. Well, we didn't know that unless we really had the intel coming out of the genomic analysis. Here, we're seeing mutations occur that cause, say, for instance, this virus to become now a dominant virus in dairy cattle operations. We didn't know what was happening until we actually did the genomics to kind of parse out to say, oh, is this mutation led to these components? We really need to take this on seriously and bring surveillance into the 21st century.
It's so tough to sample wild animal populations of any sort. Can we use, for instance, air collection or eDNA in soil samples, or what else could we do there?
Yeah, great question. So not just testing the animals, we really can do other things to test in the environment. There are mechanisms for testing air, such as an air inside of poultry houses, which, that's a pretty confined environment, and airborne pathogens can spread pretty readily. Those aren't in place everywhere, and it's pretty rare that they are tested, but it can be done. And we've seen some examples published on that. One of the other very interesting ways is testing water and wastewater. So we know human wastewater, we look for all sorts of viruses. In fact, we've been doing that for H5N1 now for a while to see if you have a human case pop up in a certain location where you otherwise really wouldn't be looking for it. And we have seen that it shows up in the wastewater shed in areas where you've had human infections as well as bird infections. And we're realizing that people are, in a lot of cases, are disposing of material into the wastewater shed that actually ends up showing up positive in wastewater, and can't tell if it's human versus bird. So we're trying to do more ways to be more precise about that. But also, there's a lot of these retention ponds and runoff ponds around dairy operations, around poultry operations. I think those should all be tested regularly just to see what's showing up. If nothing else, to provide the agriculture operation eyes on what's happening locally, but really should be done for population health purposes.
Fascinating.
Okay, we get a positive test. What's the response? How do you deal with that? And are we prepared to deal with the positive testing?
So really thinking about say the local, the statewide, and the federal responses to H5N1, dominantly, both at the state as well as at the federal level, this has been an agriculture issue, right? Because of the great impact on poultry and on cattle, and it's not really been a public health issue, and not and it's it's been important to Fish and Wildlife, but they don't really have a lot of regulatory authority. So the regulatory authority's really been on the food side, which has all been really USDA. And so if we're going to try to address the issue where you actually have transmission between agricultural operations, you probably need to look at what are the regulatory policies in place, as well as the capabilities to enforce those, but enact them. Meaning that do we have really good testing and surveillance that would help us prevent the transmission from dairy to dairy or from chicken house to chicken house? In many cases, we probably don't. It doesn't mean that there aren't a lot of great folks out there trying. It means that we're hampered probably not just by policies, but I think by political culture. Like what are we willing to accept as necessary for us to have better intelligence on what's circulating out there that can put us at risk, and especially our agricultural animals, as well as what are kind of the regulatory authorities that we want to put in place. When we talk about public health, from what's really interesting, and we saw different versions of this during the pandemic, but we don't have any great federal public health authority. The authority all rests at the state level because of the Constitution says that essentially what's not already in the Constitution means that the powers reside at the state. And when it comes to public health, it's at the state level. So it's hard to have a complete nationwide strategy for testing and surveillance purposes, as well as even response purposes when it comes to human health. And we saw that change a little bit during the pandemic, and probably trying to push a more federal response under emergency powers, and a lot of pushback. We also saw a lot of distrust coming from public health and distrust of federal authority overall. So it makes it very difficult to actually respond to our next emergencies because of how we handled ones in the past. That's something that we're going to have to address on a larger scale outside of H5N1. And hopefully we can really start to bring that back into the discussion, but I fear that's probably not going to happen for at least a while.
Thank you, Dave. That was well stated. Dave, very interesting that you mentioned about the possibility of doing surveillance in retention ponds or runoff ponds that are often associated with dairy or swine or poultry operations. What's involved in that? Do we have the tools that we need, or are there better testing tools that would enable that more effectively, more efficiently, more cheaply?
Yeah, I think what we need is, again, a commitment and probably a culture shift that to think about the agricultural business as part of the public health system, in that these are really great sources for us to understand what's moving, not just through our food, but around communities, as well as what's moving across the country. And so we don't really have a great system put in place, but the technology is certainly there. We can definitely test these types of sources. It's how do we get the buy-in of agriculture? How do they maximize benefit from that? What's their value proposition? How do we bring them to the table? And then how do we really build up a unified surveillance system with that? We have seen that runoff and retention ponds, other water sources around agriculture, are really a great source of information around pathogens that are actually being spread. We can find E. coli 0157. We can find other things. That could be just really useful information for the agriculture business. Let's try to work with them to get them better information so they can make better decisions, as well as then increase our overall intel on a more global level.
Thank you.
I was investigating in 0157 outbreak that in kids that was near a dairy farm, and public health, they couldn't get any cooperation from the dairy farm to do testing, and where they didn't have the authority to do that. And then maybe or maybe not, under the cover of darkness, I snuck on-prem and actually grabbed up samples from a runoff water source just so we could do testing, but realized that was actually fairly hazardous to do. Didn't actually go onto the property, but with the adjacent property that had this runoff water. We actually found all sorts of E. coli, but we didn't find what we were necessarily looking for. But it just meant we don't have a great way of actually doing this kind of surveillance, because we don't have partnerships with the people that we need to have partnerships with.
Yeah. Well, when it costs $1,000 a cow to cull a cow, you're not going to get a lot of cooperation without some sort of program to compensate.
Exactly.
Yep.
Yeah. Yeah. What is their value in doing that? Yep.
Exactly. You mentioned the surveillance of these ponds. Will that work for ag viruses, potato virus, tobacco mosaic virus? Will those also leach through the soils and wind up in these ponds that you see everywhere?
Yeah, there are a number of agricultural research programs. We have one here in a couple here in Arizona that we've interacted with. They're doing exactly this type of analysis to help them identify different pathogens, which may be just agriculture pathogens. And that is of use directly to the farmer or the ag operator to get that information. Do we have a blight going on? Do we have something else? It's when it has something that has an impact
Mm.
outside their own operations that they become very concerned about testing for. So it's all right now, it's all really about relationship, one-on-one relationships and working with um, these producers rather than just trying to be a regulatory body and forcing them to do things.
Dave, that's very interesting. We know in the crop agriculture side of things and the introduction of what's being called these days as precision ag, that there is quite a bit that can be done locally, even at the individual plant level, to control the growing environment for those plants. Is it possible to incorporate, the sort of wastewater surveillance you're talking about for viruses and pathogens that impact plants and automatically, feed that back into the network that these ag farmers are already using to raise their crops? And if yes, sorry for the long question, might there be some lessons there for the cattle and the animal ag industries?
I love that question, Rich, and the concept, the idea of precision ag, as we also talk about precision health, which means that we have the technologies, we have new analytical capabilities, we have an ability to be much more precise about the problems that we have to deal with, whether it's the health of our individual patients, our communities, our animals, or even our agricultural fields. And so, yeah, I think there are a lot of lessons to be learned across all of these that we could really improve because we're being more precise about what is the problem so we can be much more directive about actual interventions and solutions. So I think it's a great concept.
One of the amazing things to me about wastewater surveillance is that legally, once you've gotten rid of that wastewater, it's no longer yours, and you can't say that you don't want it tested. Um, and I just wonder if we're looking at runoff and we're looking at air collection or whatever, you really don't need the permission of the people in the area to do that. Would that make any difference in terms of the ability to do the surveillance you want?
Yeah, I can't comment about what's legal or not, what's surreptitious sampling or what is freely available or private, because I think there's a lot of different factors there. But I do think
Yeah.
that I think what you're pointing to is there a way to actually just get great information on samples that are really more freely available and that we don't have to have new laws or new regulations around. And I think that is part of what we're doing. We always want to be ethical and we want to understand what is the ramifications of actually doing testing and getting information, and then also putting information out there. We could inadvertently have a tremendous negative impact on an agriculture operation by inappropriately testing or releasing information. So it does have to be done
Yeah.
with a full knowledge about what would the impacts be. But I do think that more and more there are ways for us to do testing in the environment around us that isn't going to be concerning about whether or not it belongs to one person or another.
Right. For instance, wastewater, you don't go try to find out whose house it came from. It's part of a population surveillance.
Correct. And you have to work then with typically the municipality and their wastewater system because they control it, or sometimes it's a private system, but it's still there. What you don't want to do, and this would be unethical, is to pop the manholes right outside of individual houses so that you could understand what's going on within that one house. And that those questions came up during COVID and sampling, uh, but the technology would allow you to do it. So I think that there are some interesting ethical considerations here too.
For sure.
Fair enough.
We do have a stockpile of antivirals for flu in the United States in the event of a pandemic. I do believe it's still oseltamivir. Would that be useful in a pandemic of avian flu? And if not, would there be something else we should be doing?
Yeah, well, the good news is that oseltamivir and the similar antivirals for influenza, for human influenza, actually seem to work really well with the avian flu strains that we do see circulating. And so anytime there's a human case, they try to get them the antivirals right away to help lessen the potential for a severe infection. So it does seem to be responding to those antivirals, and stockpiles of that probably will make a difference. If it's something very large, like a COVID type pandemic, you need very large stockpiles, which we don't have that. But at least on a short term emergency basis, that is good news. Thankfully, we're good on that side of the technology. Occasionally, we do see resistance to these antivirals. We see it in the human flu viruses. So we unfortunately have to take that off the shelf. There are some other appropriate antivirals that can be used out there that are a little more rare. But we do have some things there. And when we think about vaccines, we also stockpile vaccines as part of the national stockpile. We actually do have a stockpile of H5 vaccines, but all of those are really generated against the H5 strains that were circulating in Asia prior to this pandemic strain that we've seen circulating among, or this panzootic strain, I should say, circulating among birds over the last couple of years, especially here in North America. We don't actually know how well those will work. There's probably some efficacy from those, but it's kind of like your old out-of-date flu vaccine doesn't work with your current strain very well. Same thing here. So we do need to have improved vaccines. It's such a touchy topic these days, and the technologies that can be used for vaccines, we know things like the mRNA vaccines can be quickly adjusted and adapted, but we've also kind of get past the hump of, all the PTSD, if you will, from the pandemic to allow us to move science forward to make an impact on our emergencies in the future.
This episode of Halteres Presents is brought to you by the UC San Diego Division of Extended Studies, where courses are designed for professionals looking to deepen their expertise in the biotech space. Their core course, In Vitro Diagnostics Product Development, offers a practical end-to-end look at taking an IVD product from concept to regulatory approval. You'll learn about product requirements, assay development, instrumentation, risk analysis, and key clinical and regulatory considerations. You can also broaden your skills with their Precision Medicine and Companion Diagnostics course, which covers biomarker-driven development, the co-development of companion diagnostics and therapeutics, and the expanding role of precision medicine. Ready to get started? Use our exclusive discount code HALTERES10 at checkout for 10% off the price of your enrollment. That's code HALTERES10 at checkout for 10% off. For links and to learn more, scroll down to the show notes for this episode. Advance your diagnostics career with UC San Diego Extended Studies. Your next step in biotech starts here.
I read a very interesting article over the weekend from an AI group coming out of South Africa that is using input from the Zulu Nation and other there to help teach algorithms in non-Western or Silicon Valley driven approaches to interpreting and understanding data across any variety of subjects. And you mentioned now, algorithms and that sort of thing. And I know, you guys have a huge input down there from NAU as well. There's some relationships there. Anyway, it was just a thought, is that sort of thing being incorporated here so that there's other viewpoints in the learning, the teaching?
Yeah, so just really quickly on that, I've been party to some conversations where there's a hope to build some American Indian referenced AI tools, large language models that really incorporate the culture of the various American Indian tribal communities, especially at least the discussions around here in the Southwest, so that health issues primarily can be better interpreted through a lens of the culture of the groups that we're working with. So it's kind of it's the reverse a little bit of what you're saying, but I think that it's an important step forward. We know in the sciences, when we generate large data sets, they're often lacking in many of the populations that we aim to serve. Like a lot of our genomic data, human genome data, we know next to nothing about American Indian and indigenous populations when looking at variants that may be associated with different health outcomes. That's being rectified, but it's hard because of building trust and all of that to better include
Yeah.
them in these types of studies. But the same is true when we're analyzing data. We need to be thinking about it also from the cultural perspective of the population. We can actually have a better interpretation and better knowledge experiences.
Yeah. Yeah. It's very interesting.
As I've gone to those NAU research symposia, every year for the last several years, I've had an opportunity to talk with a number of the Native American kids who are presenting posters, often on this topic, health issues in their population, communication strategies, etc. Oh, I've been so impressed. They are just so passionate and knowledgeable, and they all want to go back and help their families, and it's
Mm.
really impressive. I don't think there's anything else like it.
It's a very, very simple example, Dave, and not certainly anything that would come out of all the extensive modeling you're doing. But my daughter, of all people, pointed out to me that in Rwanda, the color purple is part of the lifeline. It's tradition, it's culture, it's generation to generation, it's past, present, future, it's
Mm.
trust. And in medical clinics, where the color purple simply has been integrated, even if it's just a stripe on the wall, is enough to change the acceptance and trust in what's being delivered. That simple little thing there could hugely color impact and outcomes research,
Yeah,
because it was just a lack of understanding of, pushing these models. Anyway, I don't mean to go down this pathway. I find it
brilliant. Yeah,
fascinating, just fascinating.
brilliant. Yeah,
Well, that was great conversation as usual.
that was a lot of fun.
Well, this is the second time that we've had you on. It's always extremely informative. Thank you so much.
Thank you so very much.
Halteres Presents is produced by Halteres Associates, a US-based bioscience consultancy that provides strategic and tactical services in the areas of diagnostics, medical devices, and life science research to clients of all sizes. The views, information, or opinions expressed during the episode are solely those of the individuals involved, and Halteres Associates is not responsible for any errors or omissions or for the results obtained from the use of this information. The information provided in this episode is for informational or educational purposes only and is not intended or implied to be a substitute for professional medical advice, diagnosis, or treatment. Halteres Associates would like to say thank you to this episode's guest or guests, and thank you for listening to this episode of Halteres Presents.